Biology 101, Lecture 14. Hello, students. We're right up there on number 14, and we're about to finish up the class. Yay! We're going to take a different approach this time. We've talked about all these really, really complicated, precise things, but we didn't quite discuss how did we get to know all this? So there's some technology behind all the stuff that I've been telling you. And we're going to look at a little bit at the technology, not a lot, but just so we know that these are certain things that are around that actually do this work. Biotechnology is the manipulation of organisms or their components to make useful products. The applications of DNA technology affect everything from agriculture to criminal law to medical research and everything in between. It simply exploits the principle of complementary base pairing to find the complete nucleotide sequence of a gene, or DNA sequencing, and that's where the beauty is. DNA is cut into fragments. Each fragment is sequenced, and the fragments, very tiny, are amplified by something called PCR, or polymerase chain reaction. That's why we call it PCR. It's too much of a mouthful. Or if we don't want to cut the DNA up, we can sequence the entire DNA molecule. Or if we don't want to do that, we have a third choice. We can take a strand of DNA and it can be sent through a nanopore, and base by base, we can detect the change in the electrical current and we'll know what the bases are in that sequence. So here we are. There are techniques, and they're right here. So here is the most famous technique, the dideoxy chain termination method for sequencing DNA. You have a DNA template, you have a primer, and then you have deoxyribonucleotides, and you also have dideoxyribonucleotides, which are fluorescently tagged. So the idea is you keep on making DNA template from the DNA template strand, label strands, and they're always one smaller. And the cap is the fluorescently tagged one. And so then you move them, you look at them, you shine a laser, and in the detector, what you will see is the longest label strand, the shortest label strand, and the last strand is always the longest one. And so you'll look at it and you'll find out what the sequence is of your DNA. Um, a little bit more advanced on this is the next generation sequencing, where you fragment the DNA, you isolate it with a bead, and using PCR, about 1 million copies of each fragment are made, and each are attached to the bead by the 5' end, and then the bead is placed into a well with DNA polymerases and primers, and a solution of the 4 nucleotides is added to all the wells and then washed off, and the entire process is repeated. And the nucleotide is, if it's joined to a growing strand, um, what you ultimately get is a flash of light that's recorded. But if it's not joined or it's not complementary, then there's no flash. So what you're looking at is there a flash or is there not a flash? And then and this is repeated until every fragment has a complete complementary strand. And so the pattern of flashes will reveal the sequence. That's one way. You can clone a gene, and if you clone that, then you would know what your DNA sequence is. So usually we do that in bacteria, and we can add it on to his bacteria DNA, and then we make copies of the gene, and we can do basic research on that. Here is the polymerase chain reaction that I was talking about and saying it's just easier to say PCR. You have a sequence of DNA, and what you do is you denature it first, so you separate it. Then you anneal it, which means you join it up with some primers, and you extend it by adding new nucleotides. And so after the first cycle, you get two um, of the original DNA sequence, two molecules of those. But after the cycle 2, you get 4 molecules. And after cycle 3, you get 8 molecules that are matching the target sequence. So you keep on amplifying how much DNA you have. You can clone genes. Genes are a tiny fragment of the chromosome. Cloning them just makes many, many copies. Production of multiple copies of a single gene is called gene cloning. We can either amplify genes by making more copies or express it by making proteins. Cloning requires restriction enzymes which snip DNA at certain precise locations, giving restriction fragments, which can be separated and visualized by gel electrophoresis. Gel electrophoresis is carried out to separate and visualize fragments of DNA. What is that? It's a polymer gel, and it's on a plastic plate, and it separates the nucleic acids based on the size or the charge or any other physical property that we're studying at that time. A DNA is negatively charged, and so when this gel is placed in a buffer, the current is turned on, so the DNA molecules are going to start moving towards the positive electrode. But the shorter fragments of the DNA will move faster through the gel than the longer fragments. And so then you read it against um, a library and you find out what you have. Gel electrophoresis is pretty good. It's pretty good because you don't really need a lot of DNA material. You only need about 15 nucleotides. But it's not as accurate as cloning. However, gel electrophoresis is really fast. It's used for crime scenes, for fossils, just one cell. It's pretty cool. So this is what I was describing, that the DNA runs towards the positive electrode from the wells, and in the end, you end up with this pattern of fragments that we can look at under certain lights, and you can visualize them. There is a systems approach to all this. Genes act together to maintain a functioning organism. Large groups of genes, when we talk about them, then we are going to look at the systems approach. This tells us which genes are transcribed and where and when. For systems approaches, we usually use DNA microarray assays. Another really good assay or biotechnology technique is reverse transcriptase polymerase chain reaction. So that's RT-PCR, and it's useful for comparing amounts of specific messenger RNAs in several samples at the same time. Reverse transcriptase is added to messenger RNA to make complementary DNA, which serves as a template for PCR amplification of the gene of interest. And then the products are run on a gel, and the messenger RNA of interest is identified. And this is making complementary DNA. And here is the reverse transcriptase PCR analysis of the expression of a single gene. Differentiation. Okay, so we're done talking a little bit about the technology. We're going to talk about other things like cells and what makes them different. Plant cells can de-differentiate, so they're called totipotent, because they can make an entirely new organism from, just a cell. Like if you have a cutting, you can make a whole new plant from that. So they can de-differentiate and they can make a new organism. So they're called totipotent cells. Stem cells, which are in animals, are pluripotent. So they can't de-differentiate. These are undifferentiated cells, but they can differentiate into many different cell types. So here is a carrot, and it has totipotent cells. You take the cells from the carrot, and you can make more plants. Whereas in stem cells, what you have is a precursor cell, and you can coax it into becoming different kinds of cells. This was actually used because uh, of the interest in cloning. And the very earliest cloning that we did on multicellular eukaryotes, and especially in mammals, was on the sheep called Dolly, where a regular cell, not a reproductive cell, but a regular cell, which is already differentiated, was taken from a sheep donor, and it was implanted into an ovary, whose ovary, an egg cell inside an ovary, where the nucleus was removed from that egg cell, and the two cells were then fused, um, and it was cultured, and it was grown, and you got an embryo, and then the embryo was then implanted in the uterus of yet another sheep. And ultimately, you got a baby sheep, which was called Dolly, which is genetically identical to the donor sheep. So Dolly didn't live very long because they found that, yes, that's true, you can maybe somehow do this, but then there are several problems. Complete genome sequences do exist for a human, a chimpanzee, E. coli, yeast, corn, fruit fly, house mouse, and the rhesus monkey. And when we compare genomes, we can have big insights into evolution and other biological processes. Genomics is the study of whole sets of genes and their interactions. Bioinformatics is the application of computational methods to the storage and analysis of biological data. Bioinformatics resources are provided by a number of sources: the National Library of Medicine, the National Institute of Health. They both joined together and they created the National Center for Biotechnology Information, NCBI. And then, of course, there's the European Molecular Biology Lab, which is EMBL. GenBank, which is the NCBI database of sequences, doubles its data approximately every 18 months. So a lot of work is being doing on this. And software is available that allows online visitors to search GenBanks for matches to a specific DNA sequence or a predicted protein sequence or common stretches of amino acids in a protein. The NCBI website also provides 3D views of protein structures that have been determined, and it's beautiful. It's very pretty to look at, and with your mouse you can manipulate the protein. You can look at it from all sides, underneath, over, above, like here. Here is an example. Proteomics. What is proteomics? Well, it's the systematic study of full protein sets encoded by a genome, because ultimately it is the proteins, not the genes, that carry out most of the activities of the cell. Genomes of most bacteria and archaea range from about 1 to 6 million base pairs. Genomes of eukaryotes are usually larger because eukaryotes are usually bigger. Most plants and animals have genomes greater than 100 million base pairs. Humans have 3,000 million base pairs. Within each domain, there's no systematic relationship between genome size and phenotype, and the number of genes is not correlated to the genome size. So there's a lot to be understood from this information. Here we have genome sizes and the estimated number of genes. So if we start from the lowly bacteria, E. coli, the number of genes it has is 4,000. And the genome size is 4.6 for haploid. So the genes per million base pair is 950. Then you proceed from there and you see that the nematode has 20,000 genes, and then we go down to Homo sapiens, and what do we see? Less than 21,000 genes. So we're no better in gene number than a lowly earthworm. So it seems. So rice, however, notice 42,000 genes. That's a lot of genes. And yet, it's only rice, and I can eat it, and it can't run away from me. So we don't know how to say who's smarter. It's certainly not the genome size. Free living bacteria and archaea have about 1,500 to 7,500 genes. Unicellular fungi have about 5,000 genes. Multicellular eukaryotes have at least 40,000 genes, or up to. The nematode, C. elegans, has 100 million base pairs, or about 20,000 genes. Drosophila has about 14,000 genes, and the human genome has about 21,000 genes, which was a big surprise when they sequenced the human genome. Vertebrate genomes can produce more than one polypeptide per gene because of alternative splicing of RNA transcripts, which is very cool because you can reuse that DNA sequence and just start at a different place, or you splice things up and you can have a completely different protein. Here is a genome size summary. Whoopsie. Um, Humans and other mammals have the lowest gene density, or the number of genes in a given length of DNA. And as we know, 98.5% of the human genome does not code for proteins, ribosomal RNA, or transfer RNA. So all that is coded or used is 1.5% of the human genome. That's what does the work. About 1/4 of the human genome codes for introns and regulatory sequences. Multicellular eukaryotes have many introns within genes and a large amount of non-coding DNA between genes. So although it doesn't code for proteins, it does do something else. We are still looking as to what that else is. Genomes of humans, rats, and mice show high sequence conservation for about 500 non-coding regions. So we're not talking about the coding regions, which would express proteins, and so we could say, well, all these animals have the same proteins. But no, these are the non-coding regions, and those are highly conserved. So about 15% of the human genome consists of duplication of long sequences of DNA from one location to another. So this is very strange. Why is it duplicated a lot? In contrast, simple sequence DNA contains many copies of tandemly repeated short sequences. A series of repeating units of 2 to 5 nucleotides is called a short tandem repeat, or STR. The repeat number for STRs can vary among sites within a genome or in individuals. Simple sequence DNA is common in centromeres and telomeres, where it plays structural roles in the chromosome. The earliest forms of life likely had only those genes necessary for survival and reproduction, and the size of genomes has increased over evolutionary time, with the extra genetic material providing raw material for gene diversification. The basis of change at the genomic level is mutation, which underlies much of genome evolution. Minor differences in gene sequence or regulation can result in striking differences in form. Accidents in meiosis can lead to one or more extra sets of chromosomes, which is known as polyploidy, as we already know. The genes in one or more of the extra sets can diverge by accumulating mutations. These variations may persist if the organism carrying them survives and reproduces, and in this way, genes with novel functions can evolve. Humans have 23 pairs of chromosomes. Chimpanzees have 24 pairs. Following the divergence of humans and chimpanzees from a common ancestor, 2 ancestral chromosomes fused in the human line. Duplications and inversions result from mistakes during meiotic recombination. Comparative analysis between chromosomes of humans and 7 mammalian species shows a chromosomal evolutionary history, and here it is. So if we look at human and chimpanzee chromosomes, what we see is a human chromosome on the left-hand side, a nice long one, and there's a telomere on top, and a telomere at the bottom. But in the middle, right where there's like this pinched-in area, it's a centromere sequence, and then there's a telomere-like sequence and a centromere-like sequence. But when you look at the chimpanzee chromosome, number 12 and 13, what you see is the centromere sequence is there um, at the bottom, and in 13, it's at the top. So it seems like the chimpanzee chromosome just sort of fused up together. Here is a human chromosome and a mouse chromosome. So mouse chromosome 7, 8, 16, and 17, and human chromosome number 16. And we look at these similarities, and it's amazing because you can see the same parts duplicated from 4 mouse chromosomes into 1 human chromosome. So that's pretty mousy. The rate of duplications and inversions seems to have accelerated about 100 million years ago, which coincides with the extinction of large dinosaurs and the diversification of mammals. Chromosomal rearrangements contribute to the generation of new species. Unequal crossing over during prophase 1 of meiosis can result in one chromosome with a deletion and another with a duplication of a particular region. Transposable elements can provide sites for crossover between non-sister chromatids. And here's a picture of gene duplication during to unequal crossing over. And so what you're seeing is slight misalignment, a little bit bigger on this side, a little bit smaller on that side. And then there's the crossover point, and what you end up with is 2 completely different chromosomes instead of identical ones. The duplication or positioning of exons has contributed to genome evolution. Errors in meiosis can result in an exon being duplicated on one chromosome and deleted from the homologous chromosome. In exon shuffling, errors in meiotic recombination lead to some mixing and matching of exons, either within a gene or between 2 non-allelic genes, as in here. Evolution of a new gene by exon shuffling. So here is exons being shuffled up, and you get a new gene, not in the same sequence. However, there are some very highly conserved genes. Highly conserved genes have changed very little over time. And these are really helpful because they help clarify relationships among species that diverged from each other a long time ago. Bacteria, archaea, and eukaryotes diverged from each other between 2 to 4 billion years ago. So the genes that are highly conserved can be studied in one organism, and the results can be applied to other organisms. And relationships among species can be represented by a tree-shaped diagram. And we've seen these diagrams before. These are evolutionary relationships between 3 domains of life. At the very bottom is the base of the tree, which says most recent common ancestor of all living things. We usually call it last universal common ancestor, or LUCA, and that's about 4 billion years ago. And then about 3 billion years ago, about 4 and 3.8 billion years ago, bacteria started to show up from the last universal common ancestor. But around 3 billion years ago, there was a divergence, and bacteria then started to diverge into 2 other shoots called archaea and eukarya. And if you look at the eukaryan tip right now, which is millions of years ago from today, what you see are the humans, the chimpanzees, and the mouse, and how much they're similar and how diverse they are from each other. So the mouse looks like it's pretty different because its root is pretty far away from the human. In gene similarity, yeah, so human and chimpanzee genomes differ by only 1.2% at single base pairs and only by 2.7% because of insertions and deletions. Sequencing of the bonobo genome shows that there's greater similarity in some regions between human and bonobo or chimpanzee sequences than between chimpanzee and bonobo. And as you can see, on the top is a chimpanzee, in the top right-hand corner, and underneath is a bonobo. And although they look kind of sort of similar, believe it or not, in some areas, humans and bonobos are more related than bonobos and chimpanzees. One of those is the vocalization gene. Humans and chimpanzees differ in the expression of the FOXXP2 gene, whose product turns on genes involved in vocalization. Differences in this FOX gene may explain why humans, but not chimpanzees, communicate by speech. The FOX gene of Neanderthals is identical to that of humans, suggesting that they may have been capable of speech. So not all that dumb. So the rates of gene evolution, a number of genes are apparently evolving faster in the human than in the chimpanzee or mouse. Among them are genes involved in defense against malaria, no surprise there, and tuberculosis, no surprise there, and one that regulates brain size, no surprise there. As a species, humans have only been around, uh, some books say 50,000, others say 200,000, but there's no way that anybody has any consensus that there were anything like humans or hominids before 200,000 years ago. Um, but they have humans have very low within species genetic variation. Variation within humans is due to single nucleotide polymorphisms, inversions, deletions, and duplications. Darwin noted that the current species are descendants of ancestral species. Evolution was defined by Darwin as descent with modification. All organisms are related through descent from an ancestor that lived in the remote past. Darwin perceived adaptation to the environment and the origin of new species as closely related processes. Evolution can be viewed as both a pattern and a process. So this graph shows descent with modification. The names that have a dagger symbol denote that those creatures are now extinct. And so this is millions of years ago. At the bottom, you can see we start this tree 60 million years ago. And at the end, which is today, all we have are elephants, which do exist, and manatees, which do exist, and then hyraxes. But everything else is kind of sort of extinct. And why is that? That is by natural selection. Natural selection is a process in which individuals with favorable inherited traits are more likely to survive and reproduce. So, observation number one, this is Darwin's. Members of a population often vary in their inherited traits. That's true. Observation number two, all species can produce more offspring than the environment can support, and many of these offspring fail to survive and reproduce. So variation in population, these are all ladybugs, and yeah, every single one of them is ever so slightly different. And overproduction of offspring, every organism does that. So here's one forming a huge spore cloud when you don't really need that many. So the inference from these two observations is inference number one. Individuals whose inherited traits give them a higher probability of surviving and reproducing in their environment tend to leave more offspring than others. That would be a logical conclusion. And inference number two, this unequal ability of individuals to survive and reproduce will lead to the accumulation of favorable traits in the population over generations, and that is true. So here is a graph, or a pictorial version of saying exactly that. The observations, the inferences that Darwin made for his theory of natural selection. Individuals with certain heritable traits survive and reproduce at a higher rate than other individuals. Natural selection increases the match between organisms and their environment over time. If an environment changes over time, natural selection may result in adaptation to these new conditions and may give rise to new species. Note that individuals do not evolve, it's populations that evolve. and they evolve over time. Natural selection can only increase or decrease heritable traits that vary in a population. Adaptations vary with different environments. Natural selection does not create new traits, but edits or selects for traits already present in the population. The current local environment determines which traits will be selected for or selected against in any specific population. Homology. So we're going to look at, we've looked at similarities, and we're going to look at similarities not in just the genes and not just the chromosomes and not just the this and that, but in another way too. Homology is similarity resulting from common ancestry. Homologous structures are anatomical resemblances that represent variations on a structural theme present in a common ancestor. So, for instance, if we look at this homologous structure, in the first picture on the left, that is the arm of a human. The next to that is the leg of a cat, and then a flipper of a whale, and then the wing of a bat. And what you see is, if you look at the first bone, which is the humerus, and it's colored in a dark blue, in the human, the cat, pretty similar. Whale, not so similar, it's very much shrunken. Bat, extremely elongated and thin. And then when you look at the radius and ulna, very similar to the cat, except the cat has more of an ulna than we do. But the whale, they're all shrunk down. And in the bat, it's all thin, as it should be. So then we look at the hand. And in the human, there are carpals and metacarpals and phalanges. Well, the cat has the same sort of structures. The phalanges are slightly bigger. But in the whale, there's a complete difference. The phalanges are enormously big, so these would be the fingers to become the flipper of the whale. And if we look at the bat, again, the same idea. The phalanges are very big. The metacarpals are really big because the bat uses its limb to fly. There are also embryological similarities. So if we look at embryos forming, the chick embryo looks very similar to a human embryo at that stage. They both have pharyngeal arches and they both have a tail. So if we look at this evolutionary tree, at every branch point, we can mark off another characteristic. So branch point, for instance, number 1, digit-bearing limbs. Number 2, there's amnion. Number 3. And so, you'll keep on going on, feathers for birds. So then you look, well, who has these things? Digit-bearing limbs, those would be amphibians, mammals, lizards, crocodiles, ostriches, hawks. So that's how you would read a tree. Those are all tetrapods. And because they have amnion, they're also amniotes. So then there's also the idea of convergent evolution. Convergent evolution is the evolution of similar or analogous features in distantly related groups. So you don't have to be very closely related, but you could have the same functionality because you use the same idea. So analogous traits arise when groups independently adapt to similar environments in similar ways. And that is called convergent evolution. But it doesn't necessarily provide any information about ancestry. So here we have the transition from of life from the sea to the land. And what you see is, again, anything with a dagger next to it is an extinct individual. So we have the living cetaceans, which are whales today, but they all came from ungulates, which are further back down in the evolutionary tree. And if you look at their limbs, You can see the humerus, the ulna, phalanges, and you can see what happened to all these bones. Biogeography. So this is the last topic. Biogeography is the scientific study of geographic distribution of species and provides evidence of evolution. About 300 million years ago, all the landmass that we have on Earth today was in the form of a supercontinent, one continent called Pangaea, which has now been split by tectonic plate movement. And so the thinking is that the species that were isolated by the tectonic drift stayed in those areas and were not allowed to go to other areas because there was a barrier of the ocean. So the ocean was a big barrier for terrestrial plants, and so um, every continent has unusual or endemic species that are local and they're not found anywhere else. Like kangaroos are only found in Australia, but not found anywhere else. And kiwi birds and so on. Um, Islands have many endemic species that are often closely related to species on the nearest mainland or island. So we do see this all the time. See you next time, students.